Hey guys, welcome back to Biopython in Python, Computational Biology. I'll just mix up the words of the title. Who cares? In this video, we're going to be continuing to work with our seq records. So we did in the last video the FASTA files, FASTA files. I never got consensus of how to say it. Yeah, so we're going to be working with more detailed files in these next ones and continuing to annotate our seq records. So let's jump right on in. All right. So now we're going to start by downloading another file. So this is a GenBank file. So I'm going to paste it here. Hopefully I'll paste it in the description. If you remind me, I definitely will. And so as you can see, this is completely different. GenBank file has a lot more information than just a FASTA file. So we're going to save this as NC 005816. See, I did it again. 005816, but we're going to save it instead as a GB file for a GenBank file. And then I'm going to upload this into the same folder that I'm working out of. So it is there. And now let's we already have seq.io open. So we do record equals seq.io dot read. And we're going to say this is the same thing as before, nc underscore 005816.gb. It should be an underscore GB, but I used a capital, so I'm going to make sure it matches. And we're going to say it's a GenBank. We're going to read it up. No such file. Oh, it's going to be gb.txt, just because of my computer being silly. All right, so we got that saved in there. And now if we type record, you can see that we have this GenBank record, so it's slightly different, but it's kind of, so if we look at before, so it's pretty similar from this view, right? But if we do something like, well, first let's do stuff that we did before. So the same way that we could look at the sequence, we can pull record seq and it'll give us the sequence just like it did for the FASTA file. Record.id, record.id. I should get a t-shirt printed that says record on it. Some merch. So you see that we have the record ID, record name, we got that, record description. So these are all the same, right? Because this was all the data that was also in those FASTA files. Well, and we can also, let's look at letter annotations. Nothing. So GenBank does not provide record annotations for their files, but we could look at, let's look at length of record annotations. So not the letter annotations, just the regular annotations. You see that we have 13. So then if we do record annotations, and then we say source, the source is where the DNA is from. So this is the actual, like the species. We could also do dbxrefs, so the reference. The project number for this on NCBI is 58037. And we can also get all the records entries in a features table. So we can do length record features, and you can see how many how many features are in this table. Okay, so what else can we do with this? So what if when genes are characterized, it's not certain that the experiment provided a clear-cut answer, right? So sometimes, for example, saying where a gene starts or ends, one research group could say it starts here, or another research group could be like, oh no, we thought it started at this location. So those are both important and relative facts for this gene, right? So what if we want to provide fuzzy information, so not concrete information about genes? We can add that as well. So you can do things like from bio import seek feature, and we can do start position, start underscore position equals seek feature after position 5. And we can do something like end position equals seek feature between position 9, left equals 8, right equals 9. Or we could do those are really, or within position. So what we're saying is, with the first one, that we think our start position for this gene feature or this genetic feature, whether it's say it could be a enhancer binding site, or a repressor site, or a methylation site, or whatever you want some DNA feature to be. We can say it starts somewhere after position 5 of this sequence, but we don't know exactly where. The end position of this feature is somewhere between 9, with the left being 8 and the right being 9, so like right in between those two nucleotides. You can also do after position, so it's kind of like, we did after position. Before position, so we could say our start position for whatever this feature is. Like I said, it could be a repressor binding site, it could be an activator, it could be an enhancer, it could be whatever. Seek feature, and we can say before position, and we'll say 10, right? And the other thing you can do, you could also do one of position, so you could do like, it's one of these two, etc. So you can annotate, and you can give ambiguous answers for this annotation too, because we just might not, we have a general idea, but not something concrete enough that we can put it in stone, and this is an important part of when you find novel genes or characterize genes, that are not, very commonly used. Like, some genes have been studied ad nauseum with, BRCA being the breast cancer gene that comes to mind, really well characterized, but maybe you identify a new gene that is associated with cancer, and you're trying to figure out some of these things. You want to provide the information as best you can for other researchers, so they don't have to redo your experiments, but it might not be set in stone yet. Okay, now that we have defined in a start position and an end position, we can create a location, right? Because we have a start and an end. So my location equals, no, not minus, equals seek feature simple location, and we'll say it's our start, which we said was before 5, and we'll say end position, which we said is between these two. Okay, so now we can print my location, and it says, okay, somewhere before 5 to between 8 and 9. We can also then pull out just the start, so we can do my location start, start, and will give us our start. Do my location end, give us our end. So we can do, if you don't want it to like print all this stuff, you can do something like this. You can say, give me just the integer for my location end. So you can see it gives you 9 instead of the description, like, you know, between, etc. Or after position. No. Sorry, my location start should print out just 5 because we said integer. So you can get rid of that stuff if you want to. Okay, so you can also just pass the numbers to the verbs if you want. So if we say exact location equals seek equals seek feature simple location, and we do 5, 9, then we can say print exact location. You can see we get 5 through 9. We could do exact location start, and it should give us 5. So you can do that as well. So start working with some other file types. So that's it for the beginning of these the sequence annotations. It's really nice for being able to keep track of your sequences, and myself up and make myself cough. So that is it for, for fuzzy locations and for some of our annotation. And we will continue in the next video doing some more annotations of these sequences. So if you are watching YouTube, please hit like, please hit subscribe, and I will catch you in the next video.